We come to the part 3 of our lecture, of our conversation, how does cell therapy work? So let's speak a bit about the mechanisms of cell therapy, of its action. But before we speak about that, we have to generally speak about aging or understand aging. So we know from various research groups that basically there are 7 more or less standard mechanisms of aging, or we call them 7 pillars of aging: disruption of the metabolism, macromolecular damage, epigenetic shifts, chronic low-grade systemic inflammation, impaired adaptation to stress, impaired proteostasis, which is the ability to excrete proteins, and of course exhausted stem cell pool and exhausted regeneration potential. You can see another explanation of these 7 pillars of aging. So, for instance, what can be done, how to maintain or how to counteract that. So if there is a cellular loss and cell atrophy, it's the replacement of that damaged cellular pool by using of the stem cells. Okay, so you can see it's on the top of the list. When we're losing cells, or cells atrophic, or they're aged, or they're dysfunctional, what can we do? We can replace them by using from a different organism, cultured or uncultured cells procured from a different organism and implanted to this affected organism to replace the damaged cellular pool. It is also important to understand the difference between the cellular senescence and quiescence. Quiescent cells are still capable of cell division when, of course, there is a mitotic stimulation, while the cellular senescent cells these are cells which are characterized with arrested proliferation potential, and unfortunately these cells are also at a higher risk of the neoplastic transformation because they continue to survive and they continue to produce so-called senescent secretory phenotype. Okay, the cells are metabolically and physiologically active. They add on to the oxidative stress, they add on to the low-grade chronic inflammation, and generally it is also characterized and described as a phenomenon SASP, senescent secretory phenotype. So cellular senescence is caused by epigenetic factors, by shortening and the erosion of telomeres, by the age-related or environmental factors related DNA damage, and by mitochondrial dysfunction. It is also caused and causes further proteostatic dysfunction. It is caused by the loss of ability to sort of eradicate metabolites, but as a result of the cellular senescence, it is also producing more of those metabolites. Hence, the dysfunction, the proteostasis, impaired proteostasis. It is also associated with nutrient signaling dysfunction very much, and the outcomes of the cellular senescence mainly are chronic inflammation, which is directly associated with practically all age-related chronic systemic disorders, and stem cell exhaustion. So, cellular senescence is characterized by cessation of cells' ability to proliferate, telomere shortening, non-telomeric DNA damage and aneuploidy, epigenetic alterations, mitochondrial dysfunction and oxidative stress, increased secretion and accumulation of various pro-inflammatory markers, and senescence-associated beta-galactosidase, very important. They are resistant to apoptosis. They are characterized by interference with cellular differentiation. Okay, that's where the dangers or the high risk of the neoplasm comes along with. They are characterized by tremendously altered, disrupted cellular function, large liposomes, and last but not the least, degradation of the extracellular matrix. So that leaves us perhaps with one and only ideal or optimal type of the cell therapy products, or the stem cells, which are precursor stem cells, are the optimal type or modality of cellular therapy that provides cellular and tissue regeneration and remodeling. And we have mentioned the subcellular products, which are cell extracts or the peptides, and they can be used and are used as a maintenance phase following the precursor stem cell transplantation, and that type of therapy provides regeneration and remodeling on the cellular level. Yeah, so we have two levels of regeneration and remodeling. We have the tissue level and we have the cellular level of regeneration and remodeling. Now, once we understand and once we have discussed the mechanisms of aging, let's see how exactly fetal precursor stem cells counteract those mechanisms. So let's specifically look into the mechanism of action of fetal precursor stem cells. Well, first of all, they do home to the site of the inflammation, and they do home to that particular organ or tissue that they correspond to or they belong to. What else? What's next? They modulate or regulate immune responses. They optimize immune responses. They reduce the low-grade systemic inflammation. They promote revascularization, because a newly regenerated tissue must be also revascularized. Yeah? So you need to regulate formation of the new small blood vessel, neovascularization, to improve and restore the supply of the oxygen and the nutrients to the freshly or the newly regenerated tissue. And by doing so, they reduce scarring, they reduce the ratio of the connective tissue or non-functional tissue or cells in that particular organ. Apart from that, in the future, they prevent the premature cell death. Yeah, so they optimize, sort of rejuvenate the entire tissue, repair and regenerate the entire organ. That's the mechanism of action. We can sort of like extract two main mechanisms of its effect. The first one is a homing effect. When the precursor stem cells, after transplantation, they do migrate and they retain in the tissues which they belong to in that particular organ, when they will further differentiate and proliferate and repair that particular tissue or the organ. And the paracrine effect. Paracrine effect is the stimulation of the regeneration and proliferation of the recipient's own cells. So it's direct replacement of the damaged cellular pool, as well as stimulation of the regeneration and proliferation of the recipient's own stem cells, whatever is still available. So you can see the different stages of this mechanism, how exactly after reaching or once being in a systemic circulation, the stem cells, due to the chemoattractors, they will first sort of adhere to the vascular wall, then there will be a transendothelial migration, then interstitial migration, and further they will home into the site of the inflammation or the site of the damage. So the homing effect is the increased ability to migrate to site of inflammation or damage under diverse pathological conditions, which makes these cells attractive for the therapy. Homing of precursor stem cells to injured or inflamed tissues depend on many signals, including growth factors, chemokines, and interleukins secreted by damaged cells and the microenvironment. And we know that such specific chemokines like CCR2, 3, and 4, they've been implicated as the factors, biological factors, sort of the chemoattractors which support cell migration. It's about the same picture, the same illustration, slightly in different way. You can see that there's a site of the inflammation, injury, or hypoxia that will produce those chemoattractants, the chemokines, various complement proteins, various bioactive lipids that will act as chemoattractors for the precursor stem cells, attracting them from the systemic circulation, promoting their migration through the endothelium of the blood vessel wall and penetration entering the tissues. We mentioned that apart from the homing effect or after the homing effect, these precursor stem cells will produce the paracrine effect, and the paracrine effect is very extremely diverse. It has the anti-fibrotic effect, it promotes angiogenesis, chemo attraction, immunomodulation, tissue proliferation, neuroprotection, hematopoietic function, and it will prevent the premature death, means the anti-apoptotic effects. And all of these effects are realized by tens or sometimes hundreds of different bioactive molecules, different growth factors. You can see just a few of them, which are mentioned here, which they perform different functions. Some of them do overlap. So all these effects are realized also not only by the homing effect, by also by very massive paracrine effect when they produce huge amount of these growth factors that will have the beneficial and stimulatory and regenerative and reparative effects on the tissues. So how fast and in which sequence these mechanisms of action are realized? The homing principle starts and happens within just the first days after implantation. We're talking about the first 48 to 72 hours are the most crucial. It was noticed that within about 5 to 7 days, the cells practically completely disappear from the implantation site, and they are spread and delivered, distributed all over the body, depending on where that type of cells belong to and where is the site of the inflammation or the injury that will express those chemoattractants, chemokines that will attract implanted cells and promote their homing effect and retaining in the affected tissues. These charts, they show the histological comparison between the xenogeneic and the human tissue. There are much more similarities than there are differences, especially in the fetal stage. So this characterization of the liver, this pancreas, the pancreatic islets, also, as you can see, is a lot of similarities anatomically, microanatomically, but interestingly that biochemically and physiologically there's even more similarities rather than differences. Same goes to certain parts of the brain, like the cerebellum as well. So to summarize this part of our conversation, I would like to make a brief summary of the unique properties of the fetal precursor stem cells based on the characterization and the mechanism of action. Let's make this short, small, intermediate summary. Well, so unique properties of fetal cells. First of all, they have high level of readiness to differentiate and to undergo various changes in response to environmental stimuli and in accordance to or with recipient's epigenome, and also able to preserve their own epigenome through generations. Second, easy adaptability due to the plasticity of tissues, which gradually decreased with each successive fetal stage and disappears at the end of the development. And these features of plasticity of tissues include growth, migration, mobility, ability to create cell-to-cell contacts and interactions. So this a very high easy adaptability is the second unique property of the precursor fetal precursor stem cells. Third one, a third unique property of fetal precursor stem cells is a highest frequency and very high speed of cell division and proliferation, which of course was sort of harnessed and improved with the new technology. A fourth unique property or fourth unique feature of fetal precursor stem cells is the highest production of various biological substances. And we're talking about growth factors, which facilitate paracrine effect. They facilitate the survival of cells after implantation and stimulate damaged or inflamed or injured tissues or just aged and deteriorated tissues and organs of the host. Fifth unique property of fetal precursor stem cells is low immunogenicity. It means that these cells, implantation of these cells, does not trigger massive immune responses in the recipient. On the contrary, it does not require a use of the immunosuppressants at all. Yeah, so it's due to its very low immunogenicity. Please remember that these are still the fetal cells that lack quite a number of those receptors that adult cells have, and those receptors and that immunogenicity is responsible for the immunological reaction and rejection of the graft, of the transplanted cellular material, and that does not apply to fetal precursor stem cells. A unique property of precursor stem cells number 6 is capability of survival on lesser amount of oxygen and when the energy is supplied by glycolysis alone. Okay, this is very, very important because that allows the cells to survive after the first minutes or hours after implantation. So once you implant them intramuscularly and before the cells migrate, before they receive the blood supply from the surrounding, that local bloodwork network, the cells will survive just because their survival ability, the capability to survive, is much, much higher compared to the adult cells, especially much higher compared to adult cells of the recipient, which usually has some kind of a condition or injury or inflammatory process.